Hello everybody, I hope you're well. This is Mr. Pettingill here. I just figured I would make a video of this lecture as a few people missed the lecture, and I figured why not just share it with everybody. So today we're going to be talking about proteins, okay? Proteins are the third macromolecule, 3 of 4, that we will be discussing in this class. We already talked about carbohydrates, glucose, glycogen, starch, etc., etc. We talked about lipids, that includes triglycerides, phospholipids, and sterols. And now we're going to talk about proteins. Now, proteins are very similar to carbohydrates and to lipids in that in the way that macromolecules can be similar, right? Protein in the food you eat is very different than carbohydrates in the food you eat, but there are similarities between carbs, lipids, and proteins. And those similarities are that just like carbs and just like lipids, proteins are made up of monomers. Okay, those monomers link together to form a polymer. Those polymers are linked together through a process called dehydration synthesis. Okay, so carbs, lipids, and proteins all have monomers. Those monomers go through a process called dehydration synthesis to become bonded together covalently. Once they're bonded together, they can form a chain of sorts and become a polymer. So the monomer of a protein is what we refer to as an amino acid. And an amino acid it's just a type of molecule, just like glucose, just like a fatty acid, just like water, just like caffeine. It's a molecule. And it just so happens that an amino acid is a specific type of molecule. There's 20 different amino acids in the human body, okay? There are a number of them, I think 4 of them, that can't be made inside of the body. And so that's why you need to have protein in your diet, because you need to be able to get amino acids into your body. It's an important nutritional aspect of your diet that you need to fulfill, particularly those 4 amino acids that can't be made inside the body. So an amino acid is identifiable by 4 different groups off of the central carbon atom that you can see here, okay? So we've got in purple the amine group, NH2. Boom, you can see that right here. Chances are if you've got a nitrogen in the molecule, in terms of the scope of this class anyway, if you've got a nitrogen in the molecule, you can kind of think protein, right? Not many molecules have nitrogen in it unless it's some sort of protein. In red, we've got a carboxylic acid group, also sometimes referred to as a carboxyl group, okay? And that's the C double bonded to an O, and that C is also bonded to an O, which is bonded to an H, okay? And we write that as COOH because this O is kind of like a separate entity to this OH. They're totally separate bonds, so they just write it that way to make it clear that it's a carboxylic acid group, something very common in molecular structure, so it's just sort of like the chemist's funny nomenclature. Don't get caught up on that. The third telltale group is a hydrogen here in blue. Boom, simple, it's just a hydrogen atom, really straightforward. And lastly, in green is this side chain, which we refer to as R. Now, there isn't an element with the atomic symbol of an R, and that's because this, much like X is used in algebra, R is used in this case to represent something that is variable, something that can change. Okay, so the side chain could be anything from another H to another carboxylic acid group, something really simple like a CH3 group, or it could be something really complicated like something that looks like a glucose molecule or whatever. And that R group, that side chain, is what is going to distinguish the 20 different amino acids from one another. All 20 amino acids have the purple amine group, the blue hydrogen, and the red carboxylic acid group, and they all have a side chain, but that side chain differs from amino acid to amino acid. You can look them up online, and they all have different, slightly different side chains, if you're curious as to see what that looks like. Okay, so also, like carbohydrates and lipids, amino acids have levels of organization. With carbohydrates, we saw that monosaccharides like glucose were linked together through dehydration synthesis to form polysaccharides like starch or cellulose, also known as fiber, or glycogen. Okay, and that was the polysaccharide. With lipids, we saw 3 fatty acids, whether it's trans, saturated, or unsaturated, would combine with a glycerol molecule to form a triglyceride. We also see phospholipids, which are 2 fatty acids combined with a phosphorus head. And we also see sterols, which we didn't really talk about the structure, but it's kind of like a honeycomb of carbon rings. Proteins are a little bit different. Proteins have monomers and polymers, okay, but the way that those monomers are organized into polymers is unique because of a process called folding, okay, and it's just as it sounds, folding, like folding your clothes, except this time you're folding proteins. And so we'll get into what that looks like and what that how you can think about that in a second. So the first level of organization in proteins, okay, we started with the monomer, an amino acid. Let's take a look at the polymer. A protein polymer is called a polypeptide. The reason for that is because a peptide is the type of bond that joins 2 amino acids. Okay, so if you see peptide, you immediately think amino acids. And if you see amino acids, you can immediately think protein. Okay, so long chains of amino acids are linked with peptide bonds. Okay, here's a long chain of amino acids. This is one amino acid. This is another. This is another. This is another. And you can see each amino acid has the 4 groups we just talked about. It's got a C. It's got the NH, in this case NH3, but don't get caught up about that. It's got the side chain. It's got the H. It's not drawn with a line to represent the bond, just for simplicity's sake. And it's got the C double bonded to a O, but you'll notice it's missing the OH because it goes through a process known as dehydration synthesis. Carbs and lipids do the same thing. It's when water is removed, dehydration, to synthesize a new molecule. So the protein packet I handed out does a good job of walking you through this, but imagine the OH of one amino acid and the H of another amino acid are removed, and in that space, a bond is made between those 2 amino acids. So you can kind of see it here. The OH that would be attached here was removed, and the other H that would be attached here was removed. Instead, we form a bond between the 2 amino acids, and that bond is a peptide bond. Okay, and then you can see that all the way through. Really recommend you check out the protein packet to solidify this concept. There's also extra practice on Schoology. So this is what's referred to as the primary structure of a protein. A polypeptide, a polymer, a chain of amino acids joined with peptide bonds. It's just a chain. It's like a bunch of paper clips linked together. Okay. The analogy that I used in class was with pipe cleaners. Obviously, I can't do that over a video, but another example might be if you think about links in a chain. Each individual link is an amino acid, and you chain them together to make a long string of links. We call that a chain, and that is sort of analogous to a polypeptide. Okay, so that's the first level. That's the primary level of protein structure. Let's take a look at the secondary level. Basically, all that is taking a chain of polypeptides and folding it on itself. Okay. What happens is there are attractions between different amino acids in that chain that cause the protein chain to fold. Those attractions are hydrogen bonds. Hydrogen bonds are a type, its own unique type of bond. It's not ionic, it's not covalent, it's hydrogen bond. We didn't really talk about in class. It's a weak bond, and it's basically just the attraction between the slightly negative oxygen and a slightly positive hydrogen. Okay, and that's why it's drawn as dotted lines, because it's not as strong as these covalent bonds. Here, it's a dotted line. Okay, and it's represented here by the yellow dotted line. And so you have the O of one amino acid getting attracted to the H of another amino acid, and it causes the protein to fold. And it's going to fold a number of times. The chain is going to fold a ton of times. And it's when it's a chain, it's not a protein yet. I said that, but it's not a protein yet. It's just a polypeptide chain. And so that chain is going to fold due to these hydrogen bond attractions. And when it folds, it's going to fold into one of 2 unique, kind of weird shapes. Okay, the first is an alpha helix. It kind of looks like one of those spiral slides at a playground, or a spring, or slinky. I guess it's kind of just like a spring, but it's got this kind of curly Q spiral staircase shape. We call that an alpha helix. You can also see it here, and it kind of points out the individual amino acids that make up the chain. The second shape that it can fold into is what's called a beta pleated sheet. This is the symbol for beta. It's a Greek letter. And this is a more unique shape. If you're confused about what it looks like, ask me in person. I've got a little pipe cleaner model that makes it clear, but it's kind of like, imagine a horseshoe, okay? It's a U shape, right? But you sort of crimp the ends so that way they're folded and kind of zigzaggy, okay? And that's a beta pleated sheet. Kind of looks like this, boop, 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 and they're parallel. And you see, again, hydrogen bonds between the different amino acids. So this is the secondary protein structure. Primary protein structure was just a chain. Secondary structure, we take that chain is folded. But it just so happens it's folded into two kind of unique shapes. Okay? Now, there's a third level of protein structure. We refer to that as tertiary. And that's when you take one of these, okay, or maybe both. Maybe this is connected to this somehow. You take this alpha helix, you take that beta pleated sheet, and you fold it on itself. So imagine you take that spiral and you kind of curl it in half. Okay, again, come see me in person if you want. The proteins packet also does a good job of describing this. That would look something like this. Okay, so what we see is we have a chain of amino acids, that's primary. Then we take that chain, we fold it into alpha helixes and beta pleated sheets. That's secondary. Now tertiary, we take those alpha helixes and beta pleated sheets and we fold them again. Okay, so if you take a look here, we've got an alpha helix, and it folds. Look at that, it folded. And maybe this is a beta pleated sheet, folded here. Okay, alpha helix folded, folded, folded. So you've got this swirly thing that's folding on itself. Okay, we call this tertiary protein structure. And there are 4 different types of bonds that can result in this tertiary folding. With this, we saw hydrogen bonds. That was it. Bada bing, bada boom. Hydrogen bonds or nothing. Here, we see hydrogen bonds, H to an O, but we also see 3 other types of bonds. Okay, a disulfide bridge is pretty self-explanatory. You have a bridge made from 2 sulfide atoms, disulfide. An ionic bond, down here in green, that's something that we talked about in class extensively. We see covalent bonds here between the different C's and the H's. That's when 2 atoms share an electron. This ionic bond, we have a positive and a negative charge that are attracted to one another, and that's more kind of like a stealing the electron. So the oxygen has a negative charge, so it has sort of stolen an electron from the sodium atom. And that doesn't form a molecule, right? That forms a ionic crystal, a compound of sorts. But it is a significantly stronger bond than a hydrogen bond. Okay, so we see hydrogen, we see disulfide, and we see ionic. And lastly, we have this pink one here. That's what we call hydrophobic interactions. So if you remember from our study of the cell membrane, excuse me, I'm just plugging in my computer here. Sorry, there's noise in the background. We talked about phospholipids that have a polar phosphorus head and nonpolar lipid tails. Okay, and those polar heads were attracted to each other, and those nonpolar tails were attracted to each other. Well, guess what? This is a very similar sort of thing. Okay, we see hydrophobic means nonpolar, doesn't have a charge. C's and H's do not have charges. Okay, you need something that has like sort of a negative atmosphere to it, and that's the oxygen. So we see that these 2 areas, these C's and H's and these C's and H's, are attracted to each other because they're both hydrophobic, they're both nonpolar. Okay, they both don't like water. Okay, so that's tertiary, the 3rd level of protein structure, and we have one final level of protein structure. It's quaternary, it's number 4. Okay, and that's basically when you take 4 of these things, and 4 different ones, different polypeptide chains that have been folded into alpha helixes and beta pleated sheets that have been then folded on themselves. You take 3 or 4 of those, maybe 5 or 6, and you combine them together. You get a blue one, a green one, a red one, and an orange one. So we have multiple tertiary polypeptides combining into one. I don't know if you guys ever watched Power Rangers, but I did a little bit growing up, and basically the Power Rangers each had their own individual sort of warrior robot that they rode in, different dinosaurs and stuff, and they would combine the 5 robots together to form the Megazord, which was like the thing to beat the big monster or something. Like they combined forces to be something more powerful. And that's kind of what we see with the proteins too. We have 3 or 4 tertiary proteins combining to form a quaternary protein that is more complex, okay, and more complicated and can probably do more complicated jobs. And the reason I say that is because we talked about earlier, structure suits function. Okay, I'll talk about that a little bit more in a second. So here's another way to visualize the protein structure, as well as an analogy that I quite like. So the primary structure is like letters in the alphabet. Okay, it's just a chain of amino acids. You can't really communicate anything with just letters. Just like you can't really do anything with a polypeptide. It's just a chain, right? It doesn't have shape yet. It doesn't have a function yet. But the secondary structure, that chain starts to get folded into pleated sheets or into alpha helixes, okay? And that's kind of like words. You can sometimes communicate things with words, just words on their own, but again, it's not enough. It's not sufficient. It's not effective enough. It's not efficient enough, I should say. So, okay, now we take a look at tertiary structures. We take those alpha helixes and stuff and we fold them on themselves. Now we start to get sentences. Math is fun. Babies are cute. I love cats. Okay, it's a bit rudimentary, but you are communicating thoughts. But if you really want to tell a story and you really want to get a job done, you got to use paragraphs. You got to write an essay. And that's why you need quaternary structure, where you're going to combine sentences together. Okay? Combining proteins together to form the quaternary structure. And the reason that this idea, primary all the way through quaternary, is so important is because we know, as we talked about, that structure is imperative to the function of the molecule. Okay? We talked about this with example was the cell wall from plants. Well, the cell wall is very rigid, and it's structured that way not out of coincidence, but because it's rigid, it serves the function of being like the skeleton helping support the plant, right? Plants don't have bones, so they need the rigid cell wall. The structure of the cell wall helps its function. And the same thing goes for proteins. The same thing goes for almost everything in life. The way it is built affects the way it functions. Okay, and you can ask Mr. TC all about that. If I'm sure he would have lots to say. But if you think about the most basic type of protein that we've discussed in class, a channel protein that's inside the cell membrane, okay, that lets water pass through, or maybe it lets ions pass through, maybe it lets something bigger pass through. It's a channel protein, a way for things to sneak in and out of the cell through the phospholipid bilayer. Okay, cool. What is that protein going to be shaped like? If it needs to let things through a layer, if you need to get through the cell membrane, what's that protein have to look like? Well, it's kind of got to look like a donut or a tube or a tunnel, right? Because something needs to pass through it, right? If it looked like a cup where one end was open and the other end was not, then nothing could pass through. It wouldn't do its job. The structure, the shape, the design of the protein affects its function. So if its structure gets messed up, if the protein doesn't fold properly and it's not shaped the proper way, it's not going to work. It's not going to perform its function. Okay? So if you were to damage a protein, if you were to unfold it somehow, which is called denaturing here, that would cause the protein to lose its function. So a great example of denaturing is when you straighten your hair or when you curl your hair. If you have curly hair and you straighten it with a straightening iron, what you're doing is you're heating up the proteins in your hair. Keratin is the protein. It's also in your fingernails. You're heating it up so much that you're denaturing the protein in your hair. You're denaturing that keratin. You're causing it to unfold. So it's losing its structure and it's losing its function. It's losing that curly attribute. And you're straightening it. And notice it doesn't last forever when you do that, right? And the same could be said of people with straight hair who curl their hair. Other things that can affect a protein's shape and can cause it to denature. I just talked about heat, but also acidity. Those are the two most common ones. But also there are salts. There are other solutions like alcohol or chloroform that could affect the protein being denatured. Something important I forgot to mention is that the order of amino acids in the chain, and like I said, there's 20 different amino acids. The order of that chain matters. It forms the identity of the chain. So, if you have a amino acid 1, 2, 3, and 4, it might form eventually form like a channel protein. But if you have 1, 2, 3, 5, it might form a completely different type of protein. Okay, so the order matters. The order dictates the identity of the protein. And so two polypeptide chains that are the same, 1, 2, 3, 4, 5, and another one that is 1, 2, 3, 4, 5, those are going to fold the same way because it's the same order, same interactions. It's going to fold the same way every time. So that's how you can reproduce polypeptides. Anyway, that was kind of an aside. But anyway, so structure matters. Structure suits function. And therefore, if you mess with the structure, you're going to mess with the function. In class, we did a think, pair, share, trying to come up with examples of proteins. This is tough. I had to look some up because I didn't have enough off the top of my dome. But here are some channel proteins we talked about. Hemoglobin's a protein in blood cells that binds to oxygen to allow oxygen to go throughout the system, the body system. Antibodies are defense proteins. Actin and myosin are what we call motor proteins in the muscle that help your muscles contract. Insulin is a signaling protein. It tells the body that there's too much sugar in the blood. People with diabetes can't make enough insulin, so they have to test their blood to see if there's too much blood sugar, and if there is, they need to inject themselves with insulin. Gluten is a protein found in plants. I'm sure you've heard of it. Keratin, as I mentioned earlier, is the protein that makes up your hair and your fingernails. And lactase is a protein that helps digest lactose, the milk sugar, the dairy sugar, and break it down into glucose and galactose. So let's talk about this is an example of a motor protein, like actin and myosin, except this one's inside most cells and it helps move things around. So this is part of the cytoskeleton, this light green thing here, and those make up the skeleton of the cell, but also help things transport around the cell because of proteins like this. This is a motor protein, this red thing, and it will attach to a vesicle or something like that, and it will carry it around by sort of binding and unbinding and then rebinding to the microtubule, and it kind of struts and walks along the microtubule and pulls the vesicle along with it. Very cool stuff. So I talked a little bit about these functions, but you can see that proteins have a wide variety of functions, from transport inside the cell to transport around the body, defense, movement, signaling, storage, structure. And this last one is one of the most crucial ones that we're going to be talking about over the next few days, enzymes. Enzymes are molecular catalysts. So a catalyst is something that speeds something up. Okay, makes things go faster. Think about like nitrous in a car, right? You have a car, it runs on gas, goes pretty fast, but you press that boost button, that speed boost, that nitrous button, your car is going to go super fast. That's what an enzyme does, but it does it on a molecular level. So what an enzyme will do is it will bind to a molecule, and we refer to the molecule as a substrate. That substrate binds with the enzyme, and the place where it binds to we refer to as the active site of the enzyme. Enzymes can bind with a wide variety of molecules as long as the substrate matches the active site pretty closely. So if you think about an enzyme as a glove A lot of people could wear the same pair of gloves, but not everybody. I could wear Dr. Alt's glove, and Dr. Alt could probably wear my glove, but there's no way I could wear a lower schooler's glove, and there's no way a lower schooler could wear my glove. It wouldn't fit, wouldn't work. So anyway, an enzyme binds to the substrate at the active site, and then the enzyme performs some action on it. In this case, it's breaking the substrate up through cleavage into two separate molecules, and then it releases them. But you could have this go in the opposite direction. You could have the blue and the yellow bind to the enzyme, and then the enzyme performs an action by causing them to bind together, and then releases it. The one that goes in this direction, from pink to blue and yellow, is a lot like what lactase does. Lactase is the enzyme that breaks down lactose, the sugar and milk, into glucose and galactose. Okay. Excuse me. And that reaction happens naturally in the body, but the less of the enzyme you have, the slower it's going to go, the longer it's going to take. So for me, I'm lactose intolerant. I have to take lactase pills. It's just pills that have lactase in it. It helps flood my stomach system with lactase. It helps dissolve the lactose that's in my stomach. If I don't take those pills, I will dissolve some of the lactose, but I won't be able to dissolve all of it. It will just be too much, and I won't have enough enzyme. So the way enzymes catalyze molecular reactions is by reducing what we call the activation energy of the reaction. The activation energy is the amount of energy required to initiate a reaction, initiate a chemical reaction. So the example we used in class was that exploding grain elevator. That gentleman used a metal shovel and caused a spark on the floor that then ignited all of the dust in the grain elevator, causing it to explode. An example we used in class that I personally really like is the turning of your key in the ignition in a car to start the chemical reaction in your engine, burning the fuel. Okay, it's the little bit of energy required to start the reaction. Enzymes reduce the activation energy. They reduce the amount of energy needed to start a reaction. So that means less time is spent finding energy to start the reaction. Right? So if you look in this top panel here, this is a depiction of what, or representation, cartoon representation of a reaction without an enzyme. Breaking down lactose without the enzyme. It's like pushing a ball up a big hill to let it fall. And you get this much energy out, but you had to put a lot of energy in just to get it started. Now you made that energy back, and then some, but you spent a lot of time pushing that ball up the hill. But with an enzyme, that hill's lower. So while it might take this guy 15 minutes to push the ball up a hill, it takes this guy 5 seconds. And then he gets the same amount of energy out, but it just happens faster because less energy is required to get things moving. Like I said in class today, imagine you're starting your car and it takes 5 seconds to turn the car over, turn over the engine. You put the key in and that's a long time for a car. But if you had an enzyme, maybe it starts right away. Boom, you just saved yourself 5 seconds. And that may not seem like a lot of time when you're driving on the road, but you get out of the driveway 5 seconds earlier. And these reactions are going to happen faster. And the faster that this enzyme does reactions, the quicker it can spit out glucose and galactose and take up another lactose molecule. And then take up another and another and another. And it just speeds the whole process up. Okay, so that's why enzymes are super important, because they catalyze so many reactions in the human body. Okay, and the one we talked about in class today was salivary amylase. Amylase is an enzyme that breaks down the sugars in starch, like in that cracker, and we saw that it breaks down the starch into different glucose molecules. So the 2 test tubes I had, the one that had just a cracker in it tested positive for starch, but the one that had the cracker and the spit in it didn't test positive because the spit broke down the starch into glucose. And if you had left the cracker in your mouth, as we did in class, you would find that over time, as your amylase dissolves the starch, it would dissolve it into glucose and it would start to taste sweeter. Okay, so this is the general idea of enzymes. And with this Apple lab, there's an enzyme that helps result in apples turning brown. And so we're going to do some experiments trying to figure out how to either prevent the oxidation or to prevent that enzyme from working, maybe by denaturing it. I wouldn't recommend using a curling iron. Not sure. And yeah, that's pretty much it. Not sure what happened there, but yeah, if you have questions about enzymes, proteins, the way things are structured, let me know. We have about a week until the test, so send me an email. Best of luck.